Welcome everyone to this week's edition of the Extracellular Vesicle Club. This is an event of the International Society for Extracellular Vesicles, and this week we are privileged to continue our collaboration with the Student Network on EVs, or SNEV, and today we have Carlos Jesus here to introduce our speaker and also to moderate the discussion later on. So I would like to just ask that everybody please put your questions and comments for our speaker in the chat box. And then at the end of the presentation, we will allow you to unmute yourselves and also to turn on your videos to interact with the speaker and generally just have a good time talking about EVs today. So Carlos, over to you.
Thank you, Ken. So hello, everyone. My name is Carlos Jesus, as Ken was introducing me. I am a PhD student at the University of Coimbra, and I'm also part of the student network on extracellular vesicles. Very briefly, the SNEV is a virtual network that that is run by students and wants to connect EV research students from all around the world. So if you are interested, just send me a message. I'm happy to show you more information, or just look for SNEV on social medias and also on our webpage. So without further delay, I'm glad to introduce Tânia Martins Marques. She is a postdoc at the Coimbra Institute for Clinical and Biomedical Research from the Faculty of Medicine of the University of Coimbra. And she's going to present how Connexin 43 is capable or is able to mediate the sorting of miRNAs into EVs. So thank you very much, Tânia, for being here and for accepting to present for this in this session. And the floor is yours.
Thank you, Carlos. First of all, I would like to thank the opportunity to be here to present this work. Just trying to share the screen. So thank you for inviting me to be here. And today I'll try to go through our paper that we recently published in Member Reports. And that addresses this question about the role of Connexin 43 in mediating the selective sorting of miRNAs into EVs. So, of course, that I don't have to tell much about this, but as you all know, extracellular vesicles mediated communication is very important in mediating a wide variety of biological processes and functions during health and disease conditions. And although EVs constitute packages of information containing multiple bioactive molecules, including proteins, lipids, and nucleic acids, miRNAs have been described as key players in mediating most of the biological functions that these vesicles exert on target cells. So given this importance, it is becoming increasingly study the mechanisms and the pathways that lead to the selective sorting of miRNAs into vesicles. And of course that many studies have already been performed, not many, probably some studies have already been performed, and some proteins have been identified to be important in this selective incorporation of miRNAs into the vesicles. Just to highlight some of them, because mostly will be important, the ones that will be important throughout this presentation. The first one that was HNRNP-A2B1 and also HNRNP-Q, also known as SYNCREP. But although these proteins have been identified and some clues have been already disclosed, there is still much that we don't know about the selective sorting of miRNAs into the vesicles. And given its complexity and the selectivity of the process, it is likely that other proteins can participate in these mechanisms, including membrane proteins. And in our lab, we have shown that Connexin 43, which is a protein that is classically associated with gap junction mediated communication, we show that this protein can be present at the surface of extracellular vesicles in the form of hexameric channels. And according to our model, these hexameric channels at the surface of extracellular vesicles can dock with hemi channels at the surface of target cells to form a fully functional channel that allows the passage of EV cargo into target cells. And in fact, we have previously demonstrated that these Connexin channels can be important to facilitate the delivery of intraluminal cargo into target cells, for example, both in vitro and in vivo. And for example, you can see here as a kind of example that EVs containing Connexin 43 are more efficient in delivering luciferin to tumor cells in vivo in comparison with EVs devoid of Connexin. So at the time, we wonder whether Connexin 43 could be used to improve the therapeutic efficacy of EVs, particularly in vivo. And that's what we were able to show, that EVs containing Connexin 43 can be used to deliver doxorubicin in vivo, and this maintains the therapeutic efficacy of the drug. Moreover, we show that when we encapsulate doxorubicin in these Connexin-containing vesicles, we were able to prevent or to ameliorate the cardiotoxicity of the drug. That's one of the major problems of these therapeutic strategies. And just as an example, you can see here that we could prevent fibrosis in the hearts of these treated animals using Connexin-containing vesicles as vehicles. So with this, we can demonstrate that Connexin, in fact, is important in the vesicles to facilitate the communication with target cells. But we also have shown, our proteomic studies have also shown that Connexin 43 can interact with several RNA binding proteins, including HNRNPA2B1 and Q, or SYNCREEP. The proteins that were previously demonstrated to be important to selectively sort microRNA into extracellular vesicles. Moreover, it was also previously demonstrated that in the sequence of the Connexin protein, there are several RNA binding motifs. Suggesting that Connexin 43 in extracellular vesicles could play other functions besides facilitating the communication with target cells. So in the context of the work that I will show you today, we hypothesized that Connexin could participate in the selective sorting of microRNAs into the vesicles. And to address this question, we started by establishing a cell-based model to study this process, namely using cells overexpressing or not Connexin 43 that will release vesicles containing or not Connexin 43. And first, we just characterize the two populations of vesicles and show that there are no significant differences in terms of concentration of particles, nor in terms of total amount of protein or RNAs. So suggesting that both populations of vesicles are similar in big terms, let's say it this way. Then we try to profile the microRNA levels, the levels of 752 microRNAs in the two populations of vesicles, so containing or not Connexin 43. And we were able to show that there are 11 microRNAs that are differentially expressed in the two populations of vesicles. And amongst the ones that are selectively enriched in Connexin 43 containing vesicles are shown in red. We can find, for example, microRNA 410, 199A, and 133B that will be important throughout this presentation. We also, considering these microRNA selectively enriched in Connexin vesicles, we performed enrichment analysis in silico analysis to identify which pathways could be modulated by those microRNAs. And we found several signaling pathways, including PI3K-AKT, MAP kinases, and also autophagy that can be modulated by these microRNAs. And these pathways can be important in a wide variety of biological processes, including during disease conditions, for instance, in cancer or cardiovascular diseases. To validate these results, we next treated cells with EVs containing or not Connexin 43, so we enriched in these microRNAs, and we were able to show that, in fact, the presence of Connexin in extracellular vesicles could modulate autophagy once taken up by target cells. And this could be important to modulate, for example, cell survival. But of course, that further experiments are required to fully understand but the biological impact of these microRNAs that are enriched in connexin vesicles. We also performed a target prediction analysis considering these connexin-enriched microRNAs. And amongst the proteins, the targets, they are simultaneously regulated by 3 or more of these microRNAs. We choose these one of the hits to validate, and we focus on this ATP2A protein or circuit 2, which is a calcium channel. And in fact, when we treated cells with EVs containing connexin 43, we show that the levels of circuit 2 are significantly reduced, so suggesting that vesicles containing connexin can impact upon calcium handling in cells. But once again, we did not further explore the biological impact of these vesicles, so further studies will be required, including to understand what could be the role of these changes during pathological conditions. Next, we wanted to know exactly what drives the selective incorporation of those microRNAs into connexin-containing vesicles. And for that, we focused, or we tried to analyze the 3-dimensional structure of the microRNAs. And as we can see here, we found a pattern. So the microRNAs that are selectively incorporated in connexin-containing vesicles present a highly structured secondary structure, a more ordered structure. Whereas the microRNAs that are excluded from connexin-containing vesicles present a more linear structure. Importantly, we were able to show that connexin 43 pulls down endogenous microRNA 133b, which is one of the highly ordered microRNAs, but not microRNA 509, which is one of the more linear ones. And we did this by UV cross-linking immunoprecipitation. We also performed in vitro assays using bioaccumulative microRNAs, either by pull down or using electrophoretic mobility shift assays. And as you can see here, connexin is able to pull down with microRNA 133b, but not as much with microRNA 509. And also when we perform point mutations that affect the stability of the secondary structure of the microRNAs, in this case the 199A, we were able to decrease the interaction or the binding between connexin 43 in these microRNAs, so suggesting that in fact the secondary structure of the microRNAs is important to the binding. Next, we wanted to evaluate whether these HNRNPs could play a role in this process. And for that, we first try to validate our proteomic approaches. So the association between connexin 43 and both HNRNPs. And in fact, we were able to demonstrate that connexin interacts with both HNRNPs and also co-localizes, and we performed this in 2 different cell lines. To better understand whether HNRNPs could play a role in the binding of connexin to the microRNAs, we established a microscopy-based assay that can be used to assess protein RNA binding. And in this case, what we did is we coated beads with the soluble terminals of connexin 43, after which we incubate these beads with recombinant HNRNPA2B1 or Q. And afterwards, we incubated these beads with fluorescent microRNAs. And as we can see here, when we looked for the binding of either connexin alone or HNRNPs alone, we did not see much interaction. However, when we put everything together, so connexin and each one of the HNRNPs separately, we do see a significant binding of the microRNA. So suggesting that these proteins could be important to together to ensure binding of the microRNAs. However, in this assay, we did not find any difference regarding binding of microRNA 133b or 509. So this assay suggests that in fact connexin and HNRNPs could play a role in mediating the binding to the microRNAs. However, the selectivity of the process is still a question. Of course, that we cannot forget that this is an in vitro assay. It's a very simple assay using pure proteins. So we have to acknowledge that if other proteins play a role in this process, namely other RNA binding proteins are important for this, or other regions of Connexin 43 besides the terminals that we used here, if those players have a role in the selectivity process, we are not addressing them with this very simple assay. So finally, we wanted to address the role of these proteins, both Connexin and HNRNPs, in the release of these microRNAs into the vesicles. And for that, we looked specifically for the levels of microRNAs that are secreted in extracellular vesicles of cells expressing or not Connexin 43. We show that in the presence of Connexin 43, the secretion of microRNA 133b is promoted, as you can see here. However, when we knock down either HNRNP2B1 or HNRNPQ, we did not see any significant changes. Both for microRNA 133b that we have depicted here, and we also performed the same analysis for microRNA 410 and for microRNA 199. And despite we see an increase of secretion of these microRNAs in the presence of Connexin, the absence of either HNRNP doesn't have much of an effect. In the case of microRNA 509, we did not see any changes amongst our experimental conditions, which is in agreement with our initial profiling. And both of these results were performed in two different and independent cell lines. Then we also performed some experiments using overexpression of HNRNPs. And here the results were very similar. So in the presence of Connexin 43, we do see an increased secretion of microRNA 133b, but we did not see a further increase in the levels of microRNA 133b when we overexpressed each of the RNA binding proteins. So altogether, what our results can tell us is that, in fact, Connexin and HNRNPs may participate together in the binding of microRNAs, but probably this process is much more complex than anticipated, and further studies have to be performed to fully understand how and where or in what particular conditions can these complexes play a role. So just as a take-home message, just to highlight that our study shows that Connexin modulates the selective sorting of microRNAs into EVs, and we show a couple of microRNAs whose secretion is promoted by the presence of Connexin 43. We also show that Connexin containing EVs modulate autophagy and calcium signaling in receptor cells, and also that Connexin, this mechanism could rely on the preferential binding of Connexin 43 to specific microRNAs, particularly those that have a highly ordered secondary structure. So many questions are still open, namely the interaction between Connexin 43 and these HNRNP, so how this mechanism could participate in the selective sorting, and also the impact of disease conditions in this mechanism are also an important question that we would like to address in the future. So just a final word of acknowledgement to acknowledge all people that contributed to the work, and you for your attention. Thank you.
Thank you so much, Tânia, for this presentation. Very clear and very nice presentation. Now I will open the Q&A session, and I will ask the people that post a question on the chat box to unmute themselves and ask the question directly to Tânia, please. So we can start with the question of Charmi. Charmi, if you want, unmute yourself, please. Maybe I can ask the question directly, and you can answer, Tânia. So Charmi is asking, how did you derive the key GG pathways from the Mirna landscape?
So we used the bioinformatic tool that is called Enrich enricher, and basically we just give the tool the microRNAs that are differentially present in the vesicles, and it gives us the key EGG pathways, and also it has a function that gives you the enrichment in terms of GO terms, so gene ontology terms. So we have also these analysis. I didn't show you the images, but we also perform those analyses there in the paper.
Okay, so maybe we can move to the next question. So Marino, please, if you want to ask your question, Marino Chavez.
Yes. Hi. Thank you for the exposition. So it's the CX43 a specific marker for EVs, or is distributed in another cell structures?
Yeah, thank you for your question. Now, Connexin 43 is a plasma membrane protein. It's its main localization, or at least it was believed so for many decades. But the more recent studies have demonstrated the presence of Connexin 43 in other structures, namely within the cell, so namely mitochondria and nuclei, and also at the extracellular space within extracellular vesicles. We also were the first group to demonstrate that in 2015, and after us, other groups have also shown the presence of other Connexin family members in extracellular vesicles from a wide variety of cell types.
Now it's Rodrigo Acuña. If you want, please ask your question.
Hi, Tania. Nice talk. My question is about the Connexin 43 exosome, because the Connexin 43 is the most abundant connexin in the human tissue. And you find a homing of if it's Connexin 43 or they are randomly distributed in the body?
That's an excellent question. We are trying to address this. At the moment, I don't have an answer for you. We believe that it won't be random. And for example, in the study that we performed to use Connexin as a vehicle, Connexin vesicles as a vehicle to carry doxorubicin, what we demonstrate is that the cardiotoxicity is reduced. So suggesting that it may communicate less with heart cells, but we did not look into that yet. But it's an excellent question, and we do want to know the answer in the future. Thank you.
Okay, thank you.
We can move to Philip, or Phil A.
Yeah. That was a lovely talk, and I'm sorry I missed the first 5 minutes of the relationship to cardiac toxicity. But what other diseases or physiological processes are involved with the microRNAs that are particularly carried by Connexin?
Thank you for your question. We did not look into that directly. What we did was this enrichment analysis to predict the biological pathways or the signaling pathways that could be modulated by these microRNAs carried by Connexin vesicles. And we found mostly signaling pathways like PI3AKT, MAP kinases, autophagy. So there are really pathways related to cell survival, cell proliferation. So it could be, in theory, involved in any disease that you may think of. So at the moment, we did not explore yet that link, but it's also a thing that we want to know and to study in more detail.
Now, this is part of a family of connectins.
Yes.
And do they go all the way back in phylogeny? Do all species have connectins?
Yeah. For example, in mammals, they exist. They are very well conserved, and in lower developed animals, for instance, in C. elegans, there are related proteins called innexins And there are also other proteins that are very, very similar that are also expressed in mammals that are the Connexin family. So we have some proteins that are similar, but in these lower organisms like the Drosophila and C. elegans, we don't have connexins, but we have these related proteins.
As far as uptake by target cells, are there molecules that have a particular receptivity for connectin?
We think that is the connexin itself in the target cell, because connexin can either exist in the hemichannel form, so it's able to dock with, for example, the hemichannel that comes with the vesicles, and we can have fully formed gap junctions at the plasma membrane. So those ones could not be receptive to the communication with the connexin vesicles, but the hemichannel form would be. So it can, our model is that the hemichannels at the vesicle surface could dock with the hemichannels present at the cell surface. And it's, at least inside the cells, you can have mixed hemichannels. So for instance, hemichannels formed with connexin 43 can dock with hemichannels formed with other connexins, for example. There are some exceptions, but general concept is this.
Last question, do the bacterial outer membrane vesicles have connectins?
Bacteria doesn't express connexin. I think they have some sort of channels that could play similar roles as gap junctions. So it's important for quorum sensing, for instance. But I think, as far as I know, anyone looked into that. So I don't really know, but it's possible, I would say.
You have many lifetimes ahead of all the things there are to do.
Yeah, sure.
It's like discovering a new planet.
Yeah.
Thank you.
Thank you.
So thank you for your questions, Philip. So Marino has another question. Please, Marino.
Yes, it's about if you have an hypothesis about why miR-133 is carried by this connexin and why not the 509 is not transported by this connexin.
We think that's a matter of sequence, structure, pardon, not sequence, because at least the 5 microRNAs that we found enriched in the connexin vesicles, all of them have this common factor, that is, they are highly structured, highly ordered. In the case of the microRNAs that are excluded from these vesicles, such as the microRNA 509, these ones are more linear. So for some reason, we didn't explore in detail the mechanism, but for some reason we believe that this highly ordered structure is important for connexin to recognize these microRNAs.
Okay, thank you.
Thank you.
Okay, so we can continue with Rebecca.
Yes, thank you for the presentation. I was wondering if you had an estimate about the number or the proportion of your vesicles that contain connexin 43.
No, thank you for your question. No, we don't have an idea of that. We didn't look into that. Sorry.
Okay, thanks.
So, and we can move to Patricia. You can ask your question, please. Maybe I can ask her question. So she is asking if carrying microRNAs are conditioned with external signal. What are known?
In the case of connexin 43, anything is known. We think that at least post-translational modifications of connexin 43 that can be regulated by external signals can play a role either in the interaction between connexin 43 and RNA binding proteins, or even with the RNAs itself. For instance, during some pathological conditions, there are changes in these post-translational modifications of connexin that could, in theory, modulate the recognition of Connexin, the microRNAs by Connexin 43, but at this moment, anything is known.
Okay, so if the rest of the audience, do you have any question, please put it in the chat box.
I have an added thing. In the laboratory of Mike Chop in Detroit, they work on MSC exosome restoration in a model of stroke, and microRNA 133 is important in that work. So that certainly would be a model to make a connection with Connexin.
Yeah, probably Connexin could be important to mediate these protective effects that they see. Yeah, because for sure MSCs express, at least majority of them express Connexin 43, so they should release vesicles containing Connexin 43. So likely they carry this microRNA.
So a knockout of EV133 could be very revealing in such a model where stroke is being treated.
Yeah.
And there are many models MSC EVs are important in, so it could have a great general activity.
Yeah, it would be really interesting to look into that mechanism. Yeah, thank you. Connexin 43 is the most expressed connexin isoform. However, not all cells express Connexin 43. So, for example, hepatocytes do not express Connexin 43, for example. So, probably you can check if your cells, like in the Human Protein Atlas or something, if they express Connexin 43. Otherwise, it's the antibodies that we used for the TEM images, we have to optimize a little bit. And there are some antibodies that worked better than others. So that also may be a question why you are not able to see. And another question is that not all EVs present Connexin at the surface. So we have double staining, let's say, for Connexin 43 and CD63 in that original paper. And there are some vesicles that have CD63, but not Connexin 43. So it's also possible that we have like an heterogeneous population and not all vesicles have Connexin.
Okay.
I don't know if that answered your question.
Okay, anyhow, I'm satisfied. And one more thing, like you have mentioned this Connexin 43 is specifically incorporate or will allow the secondary structures of RNA when compared to the primary structures or linear chains.
We just looked into the secondary structures.
So we still have time for questions, but I don't see any question in the chat box. That were not replied. But maybe I can ask just a question, a brief question. That is, do you think that using this approach, like selecting EVs with Connexin 43, could be used later as a biomarker, for instance, by checking the microRNA profile? Do you think that could be suitable depending on a disease, for instance?
I would say so, in principle. And because there are some diseases that can affect the levels of Connexin 43 in EV-producing cells, it may have an impact, a consequent impact on the microRNAs that are selectively selected to the vesicles. So probably in terms of biomarkers, it could be an important thing. I would say that. Thank you.
Okay, thank you for the answer. Well, I think we can wrap up the session. Thank you, Tânia, so much for presenting this very nice work, and to everyone for the nice discussion. And let's hope to see you on the next time. Thank you all.
Thank you.